Welcome, everyone. Today, we are going to explore one of the most fascinating and critical processes in all of biology, eukaryotic DNA replication. Imagine having to copy a massive, ancient encyclopedia perfectly, letter by letter, without a single mistake. That is exactly what your cells do every time they divide. This process is the foundation of growth, healing, and life itself. Let us dive in and see how the cell achieves this incredible feat. To begin our journey, we must understand the big picture of why and when this copying happens. Eukaryotic DNA replication takes place during the synthesis phase, or S phase, of the cell cycle. It is a semi-conservative process, meaning each new DNA molecule keeps one original parent strand and builds one brand new daughter strand. This massive undertaking is handled by a molecular machine called the replisome. Understanding this is crucial because without perfect replication, our bodies could not heal wounds, and errors in this process can lead to severe diseases like cancer. Now that we understand what replication is, let us look at a major logistical challenge the cell faces. Eukaryotic chromosomes are incredibly long, containing millions of base pairs. If the cellular machinery started at one end and simply copied straight through to the other, it would take months to copy just one chromosome. To solve this, the cell opens up the DNA at thousands of different locations all at the same time. These starting points are called origins of replication. They create replication bubbles that expand and merge, drastically speeding up the process. This incredible speed brings us to a mathematical relationship that governs biological timing. The formula shown calculates the total time required to replicate a chromosome. T represents the total time in seconds. L is the total length of the DNA molecule in base pairs. N stands for the number of active origins of replication. And R is the copying speed of the enzyme in base pairs per second. Notice the number 2 in the denominator? This is because replication moves in both directions from each origin. If the number of origins increases, the total time decreases dramatically. Before we move on, let us see this formula in action with a concrete example. Imagine a human chromosome that is 100 million base pairs long. The copying enzyme moves at 50 base pairs per second. If the cell uses 10,000 origins of replication, how long will it take? We plug our values into the formula. The denominator becomes 1 million, which represents the massive combined effort of all the enzymes working simultaneously. Dividing the total length by this combined speed gives us exactly 100 seconds. Physically, this means parallel processing reduces copying time from days to under 2 minutes. Now that we have used this formula, let us understand where it comes from conceptually. We can derive this biological equation using a simple physics principle. Time equals distance divided by speed. Here, the total distance is the length of the chromosome, L. Because replication is bidirectional, two enzymes move away from each starting point, making the speed at one origin 2 times R. When the cell activates N origins at the same time, the total effective speed of the entire system becomes 2 times N times R. Dividing the total length by this massive combined speed gives us our final formula. So we have proven how the cell copies DNA so quickly. This brings us to a fascinating mechanical challenge, the directionality problem. Imagine painting a 2-lane road where you are only allowed to paint while walking forward. DNA is like that road. Its 2 strands are anti-parallel, meaning they run in opposite directions. One runs 5' to 3', and the other runs 3' to 5'. The copying enzyme, DNA polymerase, has a strict biochemical limitation. It can only build new DNA in the 5' to 3' direction. This forces the cell to adopt a clever strategy. Because of this strict directional rule we just discussed, the two strands must be copied differently. As the DNA unwinds, one new strand can be built continuously, following the opening fork. We call this the leading strand. However, the opposite strand, called the lagging strand, must be built in tedious, backward-stepping chunks to obey the directional rule. These short, separate chunks are known as Okazaki fragments. Finally, an enzyme called DNA ligase acts like molecular glue, stitching all these fragments together. This semi-discontinuous mechanism is the only way to copy both strands at the same time. Let us visualize everything we just learned about the replication fork. On the left, you can see the parent DNA unwinding. The blue wedge at the center is DNA helicase, unzipping the double helix. Look at the top strand. The DNA polymerase is moving smoothly to the right, building the leading strand continuously. Now, look at the bottom strand. Because the enzyme must move in the opposite direction, it builds the lagging strand in short segments, the Okazaki fragments, pointing away from the fork. Finally, notice DNA ligase coming in to seal the gaps between these fragments. Now that we have seen how the main body of the chromosome is copied, we must look at what happens at the very ends. This is known as the end replication problem. DNA polymerase cannot start from scratch. It needs a temporary starting block called an RNA primer. On the lagging strand, when the replication machinery reaches the absolute tip of the linear chromosome, the final primer is removed. But there is no DNA left upstream for the enzyme to grab onto to fill that final gap. As a result, the new DNA strand ends up slightly shorter than the original. This continuous loss of DNA leads us to a profound biological consequence: aging. To protect our vital genes from being eaten away, our chromosomes have protective caps called telomeres. Think of them like the plastic tips on the ends of your shoelaces. They consist of repetitive, non-coding DNA. Because the enzyme telomerase, which rebuilds these caps, is turned off in most adult cells, our telomeres shorten every time a cell divides. This shortening acts as a strict biological clock. When telomeres become too short, the cell permanently stops dividing, a state called senescence, which is a primary reason our bodies age. This biological clock can actually be modeled mathematically. The formula shown describes the linear shortening of telomeres over time. L subscript n represents the final length of the telomere after a certain number of divisions. L subscript 0 is the initial starting length. The variable n is the number of cell divisions that have occurred. Finally, delta L represents the constant amount of DNA lost during each single replication cycle. As the number of divisions, n, increases, the subtracted portion grows larger, meaning the final telomere length steadily decreases until the cell can no longer function. Let us apply this formula to find the absolute limit of a cell's lifespan, known as the Hayflick limit. Imagine a human skin cell starts with a telomere length of 10,000 base pairs. It loses 50 base pairs per division, and it will die when the length drops to 4,000. How many times can it divide? We substitute our known values into the equation. We then rearrange the equation to isolate the term containing n, showing that 6,000 base pairs in total will be lost. Dividing by 50 gives us exactly 120 divisions. Physically, this is the maximum number of generations this cell line can survive. Now that we have solved for the Hayflick limit numerically, let us prove the general mathematical relationship for any cell's maximum generations. We start with our standard telomere shortening formula. Our goal is to isolate n. First, we add n times delta L to both sides to make it positive. Next, we subtract the final length, L subscript n, from both sides. The numerator now represents the total budget of DNA the cell is allowed to lose. Finally, we divide by delta L. This proves that the maximum number of divisions is simply the total allowable DNA loss divided by the loss per single cycle. This elegant derivation connects simple algebra directly to cellular mortality. We have covered a tremendous amount of ground today. Let us review the most important takeaways. First, eukaryotic DNA replication is a massive, semi-conservative process that absolutely relies on thousands of starting points to finish on a biological timescale. Second, because DNA strands run in opposite directions, the cell must use a semi-discontinuous mechanism, creating a smooth leading strand and a fragmented lagging strand. Finally, the inability to copy the very ends of linear chromosomes leads to telomere shortening, which acts as the fundamental biological clock dictating how we age. You now understand the master copying process of life.